Okay, now for a couple of lectures, we want to talk about formally about epigenetics. We've already talked about it when I talked about parental imprinting, right? We talked about the fact that there is signals sent down to the system from during oogenesis, spermatogenesis that control what can go on for the early stages of development. And we've also alluded to it when we talked about methylation of CG islands and things like this as part of the regulatory system. In fact, genetics is really part of regulation. That's where it fits. It's so how genes are controlled. We're not going to change the genes. We're going to change the actual way that they are modified or controlled through the system. So it's considered as a level sort of here's your genome, and there's this level above your genome that has to do with regulation. Now, it really is. I've used these little art symbols throughout the semester talking about things. We really are into the psychedelic world of the 1960s. It was a lot of fun, but it's sometimes hard to understand how these pieces fit together. But this is really where epigenetics falls. And as controversial as the 60s were, this is also as controversial in science. So we'll talk about it. But things like little examples here are my dog's eyes being different colors, or very famously, some very famous musicians' eyes being different colors and things. The fact that if we look at our palms, excuse me, our fingerprints, uh, lots of times the ones on one side, one hand, and on the other are different, right? Sometimes there are swirls on one side, or there are cones, whatever, and on the other side they may simply be carets or other forms. And in fact, they can even be different between fingers. But very commonly, for many of us, our fingerprint patterns can be different on both sides. Genetically, they're exactly the same, have the same genes underneath, but because we bud out and make these environmental things going on in the womb, right? Once the limb buds come out, they're separated from each other, and they can be in slightly different environments. You can actually end up with changes that occur on each side that are not mutations. They are simply the way there's regulatory control. Now, there can be mutations too that can change it, but there can be environmental input. So, what is it? Well, it's a field where we're talking about how genes are controlled, which genes are on, and in what ways they're on. Are they overexpressed, underexpressed, and the regulation of that form that can give you very different results in different places, right? We talked about mosaicism and things in lectures, and this can really occur in the system. Now, the term, at least the modern use of it, goes back to Waddington, who was he was an embryologist. It was in the UK, very famous. He also did paleontology, a number of things. But his use of it, and actually the modern use are a bit different, but he introduced the term back in the 1940s. And the idea was that cells would move down these sort of pathways, kind of down, troughs, and they could become all sorts of different patterns, right? Because as an embryologist, you could see that you started out with some very basic cells. Okay? And those cells would differentiate into sorts of ways. So he's talking about ways that cells would actually differentiate, and thinking about, okay, we've got these pathways, right? This epigenesis, if you will, the fact that we get this information and it spreads out into all these different organs. That's epigenesis and genetics, and he kind of put the terms together and called it epigenetics. Now, today we've taken that term a bit further, and we talk about how yes, you've got this landscape, but what happens in that process is that you've got various things that can happen to change the genetic state that you see and what can be expressed. You can have things like methylation, you can have phosphorylation, acetylation, you can have all sorts of things that can occur. And some of them are natural processes, somewhat are what drive the cells, as we talked about, in maybe neural cells. Other times they're accidental, okay, and they're forms. So let's give a definition here, and there's lots of arguments about how we'll define an epigenetic trait, but certainly it's best defined, I think, as a relatively stable heritable phenotype that's not related to changes in the DNA, but changes to the control of that DNA. Now, it may be changes in the DNA in the sense that DNA portion gets methylated, okay, or something, but it didn't change the base, right? It's not a point mutation. It's not a duplication or translocation. It's simply something physically modified the state. Or it could be even broader. Once we knew about nucleosomes, we now realize that you could methylate and demethylate, or you can do all sorts of acetylations and things to these histone proteins. And in doing that, you can lock down DNA or you can open it up. And so that's part of this whole process. Okay, and that's a good definition. It's a stably inheritable. Now, in reality, it doesn't have to be heritable to be epigenetic. Okay, so that's where there's kind of this divide. People that want it to be genetic say, well, it's got to be heritable, right? That's part of genetics. The controversy comes in when you say, well, it goes beyond that. It can be things that can happen somatically that are not changes to the DNA, but are somatic changes that aren't heritable necessarily. And so there's kind of a big divide into what, how the word is used and what people are meaning by it, which makes it somewhat difficult to discuss sometimes, right? Now, I had an early on experience with what this broad view of epigenetics is. I didn't have, of course, even know what the term was at the time or even that it was aware, but I think probably you might be able to relate to this experience. I was, when I was young, okay, I don't know, seven or eight or so, involved in sports and things and Boy Scouts or something. There was there were these two brothers, and they were identical twins. They were in fact monozygotic twins. Okay, over here, and they Gary and Gordon. And Gary and Gordon, when you first saw them, they were hard to tell apart. But within a few days, any of us could tell Gary and Gordon apart. I mean, we could see them, and if you were around them for a little bit of time, you could even recognize, without seeing them, you just hear their voices that they were different. While other people, would go into places, and other people couldn't, they call Gary Gordon or vice versa and all this, okay? And I always wondered, well, why? In fact, they were around for a few years and then they moved away. Then I saw them again. They came back when I was a teenager, and by then they were very different. I mean, you could just instantly see that they had become very, very different in personality. And I thought, oh, I wonder what, that's interesting. And when I was in college and I took my first psychology course, we actually talked about twins and fraternal twins, identical twins. And the idea in psychology was that twins tend to do one of two things. They tend to either accentuate their similarities by wearing the same clothes, doing things, so they tend to take on those persona of being the twins, or they tend to go the other direction in an extreme and become disassociated in different hairstyles, making sure they don't wear the same clothes, really separating themselves, making themselves unique and forms. And so that's kind of the ways that it was perceived. So I said, oh well, obviously they were trying, to us they were different. They were pulling themselves apart. But in reality, what was happening, and the reason we could detect them even early on, was due, I think, to what was called epigenetics. I mean, you've got these identical twins, right? They look very similar, something like this. Before too long, they start to look different, clearly different, and they're not necessarily changing their hairstyles or anything else. They're simply, their body shape is changing, and eventually they become very different. Over time, I'm sure if I could go back and see this pair of twins now, they'd be very, very different. Even anybody could probably detect differences. But even when they're 6 or 7 years old, and you've around them for a little while, you can see the differences. Okay. So what's happening? Well, remember we have this genome, right? We have this genetic material that makes up an organism, and we get these epigenetic tags. Those epigenetic tags can be, again, very much frequently they're methylation, but they can be acetylation. They can be all sorts of things that happen to that physically don't change that base. Okay, this C right here is not changed. Okay, it's simply either methylated or something happens to it, or this region in here somewhere, the nucleosomes are affected, right? And what that's called is the epigenome, okay? And the epigenome is a record of these chemical changes that occur either to histone proteins or to the DNA itself, okay? So these are sort of influences of chemical changes that occur without changing the bases or doing anything else. It's called the epigenome. Now, obviously, everybody gets the same information, right? I mean, dad, you get half of your information from dad, half your information from mom. But we've already talked about the fact that, at least in mammalian systems and many others, we send down epigenetic tags, right? We talked about genetic imprinting. There are these little tags that occur, and over time, those tags start to disappear, right? They're there for the first few replication events, the first few splittings, mitotic events go on, you start to have them. And then ultimately, though, we start to remove those tags. However, some of them stay, right? Some of them stay for long periods of time, and those become known as imprinted genes, ones that really do stay and have a long-term effect on the system. And so those are the ones that we would think about as true epigenetic control, because they're heritable, right? They're things that are going to stay that way, particularly with, say, methylation. We have, again, enzymatic systems that will once they replicate DNA, will actually transfer that methylation over. Oxidative damage and things, oxidative changes tend not to do that, tend to get corrected. You know, we're not talking about mutation here. We're talking about just having a change to the chemical structure. But the epigenetic ones will do that, okay? So we're talking about these epigenetic genes that will change, and that's really what you see, okay? You have a monozygotic twin, right? You have a single egg, embryonic. They get the same tags at first, okay? And as the embryo develops, okay, both of those develop, those tags get turned on, they get specialized, right? And what happens is that the epigenetic tags get either silenced or used. So the embryos are, just every embryo is developing based on these signals sent down from the parents that move along, okay? So that basically by birth, you should pretty much have the same information, right? And they look very much the same if they're true twins, and they do, okay? Now again, even monozygotic twins sometimes will have slightly different fingerprint patterns. You know, you say, well, that shouldn't be. Well, it is, because again, they're even in utero, you can have slightly enough environmental changes. One plays relative to the other, and you can have slight differences. But for our purposes, we'll just say that they come out with basically, you know, identical. Okay. A few years later, though, you start to get different tags in different places. They will change, those tags will change. So some will be removed, but then you'll start gaining others, okay? All right, so the tags that have stayed constant are the ones that are really traditionally been called the epigenetic tags. The new stuff that's added, I think, is epigenetic, and it's what's going to change the system as we go over time. It's what's going to make these two twins diverge more and more and more over time. Now, whether that's heritable, whether they're going to pass those differences on to their offspring or not, is debatable, but it's an interesting thing. Certainly, some of them are going to be passed on. Now, what does this? Well, what does it is environment. Okay, environmental factors. One of the biggies is things like diet. You know, your mother was right when you need to eat your veggies and you need to eat your fruits. Because if you eat things that we all love that have these fats and other things, those can cause oxidative changes. They can cause all sorts of things that go on. Physical activity most certainly can cause it. Individuals that are obese tend to have far more epigenetic tagging going on from oxidative damage than I really adipose, the fat that's around the bellies of males and upper thighs of females, and bellies as well, but usually upper thighs, is adipose is an organ, and it sends out signals. And one of the ways it sends out signals is through oxidative changes, through these free radicals. And it actually it is going to modify your brain and stuff. And that's a natural process in many mammals, because when food becomes available, right? You want to fatten up, right? You know, I was watching something the other day of bears in Alaska, and a salmon run was going on, and they're just out there pulling fat and eating as much as they possibly could, because they've got to get through the winter. Well, they've been signaled to do that. Well, it turns out our adipose does that as well, because we have lean times when there's less food available. And so when there is food, harvest time, we tend to do it. Well, in today's environment, there's food around constantly. So the system gets to overriding it. Well, part of what the signaling is oxidative signaling that comes out of our adipose, and it can signal our brains, other things. And so it's sending these signals up. And if you're perpetually obese, you tend to have lots of epigenetic modifications, many of which can be bad. They can lead to cancers and all sorts of other things. And then there's things like toxins. I've railed about smoking and that sort of thing before, and that's certainly one of them. But it also can be artificial ingredients that we find in our foods that aren't part of the natural system. And then it can be things like stress. individuals who are under heavy stress, they go out and they really have these high pressure jobs or all sorts of things, or can have emotional stress in their lives, will tend to have more epigenetic markers and epigenetic things go on than individuals who have less stressful environments. So you put all those together, okay? You put different foods, you put exercise, you put all these together. By the time you reach older age, you see very, very different types of forms, okay? So twins, by the time they reach them, one's up walking, doing things, being very active. The other one is basically almost immobile, you know? So it does matter as we go through life what we do to our systems. And so, yes, they're going to end up being unique because even though they start out genetically identical, uh, replication, true mutations that are going to occur and changes that are going to happen as we go through and we replicate our system, but we're also going to acquire these systems as well. Okay, these changes that are epigenetic. Now, interestingly, this kind of goes back to both Lamarck and Darwin. Um, the idea that this epigenesis that I said that Darwin had come up with. Well, he didn't come up with it. It had been around. I mean, goes back to the Greeks really, and certainly Aristotle sort of had these ideas of epigenesis, but the that's epi that's epigenesis, not epigenetics. Okay, and so it was these changes and these influences that came along. But Darwin said, okay, we've got this variation, there's inheritance, there's differential survival, then we get extinction. And Lamarck said, well, there's this part of it about these acquired characteristics that becomes a big influence there, and basically I've used this little argument before, but on the Lamarckian model, a giraffe's necks would change because they would stretch them and they would acquire these somehow changes to the pattern, and the tumules or things that came from that would be passed on to the progeny. Darwin said, no, you get these inherited changes that make you slightly different, and those went out. The taller ones in this case can get more food. And we see that, I mean, that's neo-Darwinianism is just taking that to the extreme. We talk about things like, Darwin's finches, right? This adaptive radiation, rapid adaptive radiation that went on, and all that came from these, these gene changes, and evolution could be very fast. And because selection, there was all these open niches out on these islands for these birds to occupy, and so they needed slight modifications. And you've read the whole stories about all of these various forms of how selection plays a role. Well, where epigenesis and epigenetics connect, okay, is through this Lamarckian idea of transmission of acquired characteristics. Now, epigenesis is not Lamarckian theory, okay? It's not. But the construct that had been thrown away for so long with neo-Darwinianism, which was that it was all about the genes, right? It was all about what you had and who you passed it on. It was gene-centric view is neo-Darwinianism, and it just said no. This transmission idea, this fact that we pick up these acquired characters, that's not the way it works, okay? But by looking at epigenesis and some of the things that come there, and the fact that it can be transmitted, has pulled us back to this idea that, yeah, environment does play a role that in a different way than you would think of it in classic neo-Darwinianism. And it's led to, I like this figure a lot, okay? This is one from experimental biology, and here's Darwinism, true Darwinism as it was put forth in the 1800s. There's variation, inheritance, and natural selection, right? As the three must-haves, right? You must have variation, you must have ability to inherit that, and then you got to have some fitness modification. And then when the neo-Darwinianism came along, we had Mendel been rediscovered. So we had gene mutation, we had Mendelian inheritance, all the ideas of population genetics had come along, and there were other things, right? Now, the outside loop here says, this is what neo-Darwinianist view is that it's DNA is the driving force, right? That's what's inherited, and therefore that's what selection works on. It's gene selection is important, and it's really, the genome is sort of an isolated unit that builds this organism. If it's a good organism, it puts on more progeny. If it's a bad organism, it puts on less progeny or something, okay? Well, the real world's over here, okay? Unfortunately, this is what's out there in most general textbooks. It's certainly the model that you'll find out there and popular literature and that sort of thing. But as biologists and people that think about this, and certainly in population genetics, this can be moved over to this side. We really think more in terms of plasticity. We think about multi-level selection. We think about selection going on where, in an idea where you have a plant and you have the next predator on that plant has a genome, modifies its genome and its allelic structure to match what it can get, maximize out of that plant, which then layers another layer. So you have these communities of selection going on. And that all has to do with evolvability and things. What we're talking about now, though, is epigenetic inheritance. And notice it's outside of all this loop over here, okay? These are little Venn diagrams, if you will. And epigenetics and neo-Darwinianism are just battles. And I've talked about battles before between individuals. When Darwin's ideas came along, we see these battles. We'll talk more about that when we get to population genetics. But there are these battles, and this is one right now. If you want to go read literature, this is 2015 here, but 2016, 17, 19, whenever you're 20, whenever you're looking at this, I guarantee you there's a battle royale going on about whether epigenetics matters or whether it's just these little changes, And the battle is about whether it's inheritable or not and what types of changes it has. And there is data that things coming from the laboratories primarily, but stress and things that are done to one generation reflects in the progeny, and then that carries through even to the next level. So there is data out there supporting that. Now, does it happen in every gene in every situation? No. Okay, so we're certainly not away from this whole gene selection level, but the epigenetics brings a whole new thought level of process to all of this, and it certainly fits in with the way we're thinking in modern genetics and in modern evolution and how things function. Okay, all right. So that's an overview, and I want to go now. The next one we'll talk a little bit more about some of these mechanisms and things that we would see in epigenetics.